thanks for the kind introduction, and thanks to Lee for organizing such a great symposium and for the invitation to speak, and happy birthday. I'm going to, actually the previous talks have set up what I'm going to speak about very nicely, and I'll try to allude back to some of the points that have been made throughout my talk. This is my first slide. I already noticed two mistakes. I got the date wrong, and I could never figure out if this should be systems chemical genetics or chemical systems genetics. Probably the former. Anyway, so what I'd like to do is actually start with a very old icon in systems biology, which I've shown here before. And this is an image, a hand-drawn image, well before the days of computers. In fact, before even the vaguest notions of the structure of the gene had been realized. This is by Conrad Waddington, who's the chair of animal genetics at the University of Edinburgh for more than 2 decades. And he really, he published a lot of his ideas in books. In this particular book called The Strategy of the Genes, he elaborated this idea of the relationship between the genome and this sort of imaginary, at that point, huge collection of genes, shown by these kind of tent pegs on the bottom, and this undulating tent or canvas surface, which he imagines as a phenotype. And so there's a couple of key points. One is that genes, the same gene connects to different points on the surface, which Ellen just told us about. And the other key point is that all of these cross connections between these guy wires represent these gene regulatory loops, and that's at every single level that occurs. And I think this is the big problem 60 years later, after this diagram was drawn, that we're starting to come to grips with. And so Trey talked about this genotype to phenotype relationship, this complex algorithm, and I'll talk a little bit more about how the environment, and in particular how many different kinds of chemicals and drugs perturb that relationship. I don't have a picture of Lee here, but I have a picture of one of his very famous early instruments, the ABI 370, which really, I think, broke open the first part of this diagram in allowing entire genomes to be sequenced and the entire sort of problem to be laid out. And that has left us with this complex legacy of figuring out how does it work. Okay, so systems genetics from yeast to humans, and there's a couple of surprising points I'll make here. So within Waddington's landscape, most of what we know has come from one eukaryotic organism. So in current databases, the interactions between genes represented as synthetic lethality or synthetic suppression or synthetic rescue. 95% of those interactions come from yeast. Okay, it's a very big jump to go from yeast to human cells, and so this is, building this network that Trey alluded to took about 15 years, and I'm guessing between $30 and $40 million in funding to build one network, really for yeast under one growth condition. So it shows how difficult it is to do this mapping and how far from being automated it really is. The other point is that although we talk about this human genetic landscape, about half of all human genes are barely annotated. We have no idea what a large fraction of the genes do. So I think with the development of CRISPR-based gene editing technologies, and in particular CRISPR-based genome-wide genetic screens, we're really just at the beginning of a tidal wave in the next very small number of years, where a lot of this information in yeast is going to be extrapolated to humans, and we're going to find out some things that are very, very different. So work from Feng Zhang and David Sabadini and Eric Lander and Jason Moffat and others really showed that this technology, which fundamentally works just by making very precise cuts in the genome, and then error-prone repair of those cuts generates indels, which very often lead to loss of gene function. This technology is so robust for screening that it actually allows you to undertake these screens and make reliable conclusions, which is quite in contrast to previous technologies. Okay, so that's by way of a bit of a long-winded introduction. All of the data I'm going to show you is using our own CRISPR libraries built that correspond to all ref-seq genes in human cells, but also all of the alternatively spliced exons we could identify, about 5,000 putative ORFs for which there's really very little information that these have any function whatsoever, as well as 2,000 non-targeting sgRNA controls. All the experiments are carried out in this cell line called NOM6, which is a pre-B cell lymphocytic cell line. It has very good properties. It grows in suspension. We can do these screens very rapidly in parallel. It has a pseudodiploid genome and about a 24-hour doubling time. So all of the data I'm going to show you actually goes back to the same infected pools of cells, and we were very careful to set this up so that we weren't hitting cells with libraries in different phases of growth. All of these derive back to the same pool. We have Cas9 under inducible control, and when we start our experiments, we thaw up a pool, we turn on Cas9, and so it's all very, very reproducible from experiment to experiment. That's important for some of the data I'm going to tell you about. And we use standard next-generation sequencing to work our screens up. And basically, all we're doing is looking for either enrichment or depletion of sgRNAs in the pool after whatever selection we apply to the cells. Okay, so in the first instance, when we were just benchmarking this system, I'll just make one point that actually, to us at least, was surprising. And that is that when we compared data across, at that point, the genome-wide screens that had been published in different cell lines and compared it to our data and organized it in this simple heat map here, there's a really striking feature that the number of genes that appear to be essential, which we call universal essentials, or genes that have a severe fitness defect when they're cause a severe fitness defect when they're disrupted, is actually very small. It's less than 500, and that's just across 10 cell lines. And in contrast, in yeast, which I forgot to mention on that first slide, yeast has about 1,100 genes that are essential for viability. So the number of essential genes, if you use a strict criteria in human cell lines, is very, very context specific. And so taking this analysis and extrapolating it further across a much larger data set of 342 cell lines, drives that number of universal essentials, so genes that are essential in all cell types, down to less than 500. Okay, and so this gets back to, I think, what Ellen alluded to in her talk, the extreme context specificity of function. And if you want, in some ways, you can think of humans as 200 different organisms that are collaborating with each other to make the whole, right? Every cell type is so different from each other. And if Rudy Aebersold were here, he would tell you about proteomic evidence that is consistent with this interpretation of the genetics. Okay, so the extrapolation from yeast to humans in the very first simplest question failed, basically. And so we think about this in this way. This is a sort of an overview of the protein interactions. These universal essentials tend to be the cores of protein complexes. There's a lot of what we call contextual essentials that are essential in some lines, not others. Those tend to interact very tightly with this universal essential core, and structurally, they're essentially the outer subunits of these protein complexes. So there's a structural and evolutionary component to this organization that the genetics has revealed. And again, there's many, many more contextual essentials than there are universal essentials. So we call this the onion model for obvious reasons. And I think this has implications for things like observations like tissue-specific syndromes, why when you mutate a particular gene, it's only manifest in certain tissues. And I think it's also very, very important in thinking about therapeutic opportunities. Just because a target may be an essential gene, it's not going to be essential in many, many different tissues. And so you have this differential window of susceptibility. Okay, so what I'd like to turn to for the next 10 minutes or so is extending this these genome-wide screens to many hundreds of different small molecules ranging from toxins to drugs to compounds of unknown mechanism of action to nutritional supplements to explore what happens when you perturb that genetic landscape. So this is I think, certainly at the time was a pretty audacious title of a review that Stuart Schreiber wrote, saying that the small molecules are the missing link in the central dogma. But I think Jim made a very, very strong case that actually you just can't treat a biological system in terms of trying to understand it without taking all of these small molecule interactions into effect. And so, the question is, how do these small molecules perturb Waddington's landscape, and how do they inform us as to the structure of that landscape? Okay, so I heard a couple of good quotes from Ralph this morning, and this is a quote from Oscar Wilde: "Quotation is a serviceable substitute for wit." In other words, if you can't be intelligent, at least quote someone who's intelligent. So I think Ralph and I are on the same page here. And so my first quote actually introduces the next speaker. He's probably going to turn red and crawl into a hole when he reads this, but I think this is a great quote from Roger Perlmutter. "Drugs fail because we don't understand biology." And so these CRISPR-based screens are a very, very good way to interrogate drug mechanisms of action. So I'm going to show you a few examples of that. Conceptually, this is nothing new. This is taking a page from the Yeast playbook. Ron Davis and Guri Guyver and Corey Nislau and their colleagues, as well as Charlie Boone, have used the yeast gene deletion collection to basically interrogate drug mechanisms of action for more than a decade now. And these screens have proved incredibly informative simply by correlating the patterns of gene deletion, sensitivity, or resistance to a particular perturbation, and then clustering those profiles to identify not only the effects of a drug on different sets of genes, but also to correlate genes across that perturbation network to infer gene function. And so this has been done at a much larger scale by Chad Myers and Charlie Boone recently. And it turns out that the chemical genetic interaction network actually mirrors the genetic interaction network quite closely. And we've used this kind of data to for example, predict synergism between different compounds. I'm not going to talk any more about that today, though. Okay, so here's our KIMA genomic profiling pipeline. We do very careful dose response curves. We like to screen our compounds in an IC30, and the reason for that is we can detect both resistance to the compound and sensitization to the compound. So in one experiment, we get both. We get the genetic interactions in both directions. We start with these pools that we then split into about 60 cultures per cycle. We grow the cells for 8 days in the presence of the drug, so about 8 population doublings, run it through our workup pipeline. And this is kind of the current summary, this huge matrix of gene-drug interactions. So we've run about 350 screens to date across all manner of different compounds, kinase inhibitors, epigenetic inhibitors, metabolic inhibitors, and so on and so on. And I'll quickly run you through some examples of these. So as I said, we take great care on these dose response curves. And one lesson that we've learned is that the shape of the dose response curve actually matters a lot. And if we've tried to run screens on compounds that have these very sharp dose response curves, and usually they fail because these are all done in 384 well culture. We extrapolate that to a 70 mil culture that we need to do for these screens, and it just doesn't scale very well if the transition is too sharp. So we tend to like these slightly flatter curves, and as I said, we do it at the IC50 value. Okay, and this is Paracelsus, who's the father of toxicology, and that's his quote: "The dose makes the poison." I'll come back to that in a second. Okay, so one thing that validated this approach is that, as you would expect, compounds that have similar mechanism of action or similar structure or the same compound run at different doses tend to cluster together. So that's good. Here's the overall matrix. You can see there's not any massive clusters, so we've got very good spread across gene function, and I'll touch on this again in a moment. Here's one of the obvious clusters that turns out to be the mitotic spindle checkpoint, and the reason for that is that we've run about a dozen different microtubule poisons, and although they all have different profiles, they cluster around this mitotic spindle checkpoint. Okay, so does this work? I'm just going to run through very quickly some data slides so you can look for your favorite gene on these slides and ask me questions at the end that I won't be able to answer. So here's trail ligand. This is a very strong selection, activates the extrinsic apoptotic pathway. For those of you who know this pathway, you'll see this single experiment basically lit up the entire pathway. We've got both activators and inhibitors of the pathway, as well as actually a number of uncharacterized genes that are probably new components of that network. We're also very interested in toxins. We collaborate with Health Canada, which is our equivalent of the FDA. I was very glad that Ralph has introduced this term, the exposome. I just put a little bit of extra data onto, a little bit of introductory background onto this slide. Mike Snyder published a paper a couple of weeks ago in Cell, really reporting the first longitudinal exposure study to the exposome. He wore a personal monitor for 5 years and collected data, and it's a very, very intriguing data set. He looked at both bacteria, fungi, viruses, and environmental chemicals. These are some of the top environmental chemicals. This one here, DEET, which you all know is an insect repellent, is ubiquitous in the environment. It's everywhere. Other compounds like diethylene glycol, a suspected carcinogen, pesticide orthomethylate, and other environmental pollutants, chemical pollutants are also very, very prevalent. So we're exposed to this stuff all the time. Here's the kind of data you can get out of this. This is for actually for arsenic, which in the developing world is a massive environmental contaminant that poisons drinking waters. And the kind of hits you get from these screens, so you have this massive, set in the middle of genes that aren't moving. And then typically these signatures, are anywhere from a few dozen to hundreds of different genes. But the ones you can really trust are the big outliers at the top and the bottom. So at the top, there's always resistance genes. At the bottom, there's always genes that cause sensitivity to the drug or the toxin. So we get things like this is a phosphate exporter. We get other genes in phosphate metabolism. So this is exactly as you would predict if arsenic is mimicking phosphate, but we also get surprising things. So these are all 3 subunits of AMPK. These are other transcriptional regulators that have not been connected to arsenic. And so this sort of lays out a whole new set of nodes in this genetic network that need to be connected to each other. Okay, and so there's many, many different examples for these toxins. Here's a particular toxin called ethanol, which some of you may be familiar with. This is run actually at 1%, but that's only 3-fold higher than the maximum tolerated dose for humans. Apparently, at least reading recent literature in the New York Times, once you get to 0.3 or 0.35%, you can have these blackout periods where you can't remember, what you did. But look at all of the genes that change here. This is really not good, and it's coincidence. I basically stopped drinking after we got this data, and so you get these apoptotic signatures. This is actually so this correlation plot is actually with an epigenetic inhibitor, which is an inhibitor of the PRMT1 methyltransferase. And so we're starting to tease out these different apoptotic responses and building patterns for that. Here's one of the very few drugs against the ubiquitin system, 26S proteasome inhibitor called bortezomib. And you can see you get these interesting patterns. So translation initiation factors actually suppress sensitivity to the drug. A lot of proteasome subunits confer sensitivity, but some of them confer resistance. And so this is another indication that these complex feedback loops can exist, and you can't necessarily predict how the system's going to respond. This is another drug that's, I think, in late-stage clinical trials. It's an inhibitor of the Wee1 cell cycle kinase. Wee1 is an inhibitor of CDK2. Here's CDK2, comes out as one of the top hits. Here's cyclin A. This is SCIP2. SCIP2 degrades P27, which inhibits CDK2. So you can see when you know the genetics, you can look at these profiles and say, hey, this all really starts to make sense. But then there are all these other interesting things that come out that are equally strong hits. These are EIF2 alpha kinases, so GCN2 and its activator GCN1. So lots and lots of new biology to figure out. This is another inhibitor that's just been recently approved by the FDA. It's a CDK4/6 kinase inhibitor. CDK4/6 are the kinases that phosphorylate RB. So you take out RB, now the cells can grow in the presence of the inhibitor quite happily. But RB is not actually the top hit. This autophagy gene called AMBER1, which is still relatively unstudied, turns out to be the top hit in these screens. We've also run many different epigenetic inhibitors, and you can see this particular inhibitor makes a cell go pretty haywire. But again, the genetics, when you have an idea of what the target is for the compound, starts to make sense. So this top hit here is the demethylase. So you suppress the loss of the methylase by deleting the demethylase, and conversely, here's a H3K9 trimethylase. When you take that out, the cells become even more sensitive. So you've eliminated one of the pathways that buffers against this compound. I'll spend a couple of more minutes talking about these microtubule poisons. So noconazole is very widely used in the lab to arrest cells in mitosis, and gratifyingly, the entire mitotic checkpoint pathway drops out. We typically arrange these hits in different orders, and one of which is to arrange all the genes by chromosome. And we saw this massive column of hits here. This is really puzzling. There's literally several hundred hits in here. And it turns out that MAD1 is right at the distal end of chromosome 7. And what these hits represent is any breakpoint between MAD1 and the centromere will cause a loss of the end of the chromosome, which gets selected for in this screen, and that's why all of these genes score as a hit. This, I think, is a piece of evidence that we better be pretty careful with CRISPR technology, because it means these chromosome breaks and translocations may be much more frequent, and particularly if they're selectable, this could be a very serious sort of hidden problem in engineering these cell lines. And one of the points I want to make is that different drugs, even if they're hitting the same target, can respond very differently. This is another microtubule poison. I forgot to write down the name on the slide. I can't remember which one it is. But what you can notice immediately is that it doesn't have that chromosome 7 column, yet it still hits most of the spindle checkpoint. And so the other point I wanted to make, sort of in this general vein, is that many compounds can have dual modes of action. This one here is rotenone, which is a well-known rat poison and electron transport chain inhibitor, and we compared rotenone at high dose and low dose, and what we can see is that there's a very nice correlation, but there's this outlier that's only manifest in the low dose, and that turns out to be the spindle-assembly checkpoint of all things. And if you go dig through the literature, you can actually find some evidence that rotenone is also a microtubule poison. And so here's a cluster of all of these different microtubule poisons and other mitotic inhibitors we've run. There's rotenone right in the middle of the cluster, and you can see there's a lot of diversity in this cluster. One of the other compounds that landed into this cluster is a compound called DAB-N that we were working on for other reasons, and it had this diagnostic chromosome 7 column of hits, and it turns out to be a previously uncharacterized mitotic or microtubule poison. So these these profiles are very informative, and I think the message I'd like to leave you with is that every drug, certainly that we've looked at, that's supposedly hitting the same target has a different genetic profile, and I think we could talk at length about what the reasons are for that, but that's the fact of the matter. And I think it comes back to Roger's quote, the reason drugs fail is we don't understand biology. I think these screens will get us towards understanding. Okay, the final point to make just on these CRISPR screens is that these screens, so you've probably all heard of this problem of study bias, where almost all publications are on a few hundred genes, and that's what's represented in the orange and green bars here. The blue bars are the data from our KIMA genomic screens, and they're really telling us a lot more about human gene function in an unbiased way. And so I think, again, this is going to be a very, very exciting next few years as we go through, and more of these screens get run, and we can start to correlate genes with each other across all of these different data sets to infer human gene function. So these are the functions from our data set, and I think that those points just summarize kind of where we're at currently. Okay, so for the last bit of the talk, I want to change gears into things that are looking further down the timeline. I wouldn't be all that comfortable talking about these in a regular symposium because I would be full of bullet holes by the time the talk was over. But since Lee wanted us to talk about vision, here's part of our vision for the next, in the next few years. So I think it's very clear that that natural products have been a tremendous source of new drugs for us. In fact, depending on how you want to split the argument, a majority of the drugs that are on the market today are actually natural product derived or natural product inspired. And the reason, in part, these compounds make such good drugs, they've been selected through evolution to interact with biological polymers to change phenotypes. And so this is part of that fourth dimension of the genetic code. And these molecules have properties, aside from their size, that make them they're less chemically tractable, and so they tend to have three-dimensional structures. So they're enriched in sp3 space, and they tend to have chemistries that are not so easy to mimic synthetically. So these are complex molecules that are selected for bioactivities. The problem is that there are many problems in natural products which have caused them to fall out of favor. One, many times you can screen for a natural product activity. You put all the effort into sourcing it, running the screen, purifying it, figuring out what it is, and it's the same thing that was discovered 30 years ago, even though it's a different organism and even though it may be made by different biosynthetic genes. So there's some kind of selection in nature to remake successful compounds, and that really messes up drug discovery. It may be that natural selection has been limited in how much of this space it can explore. The producer organisms can be hard to grow, and so all of this has sort of led to this approach being abandoned, I think rightly so, in favor of more interesting chemical synthetic approaches over the last 3 or 4 decades. But what has happened is that it's become very clear that it's possible to exploit the ever-growing knowledge of these biosynthetic pathways. And the key feature of these pathways is that they're modular. And as more and more of these pathways are understood and more metagenome sequences are dumped into sequence databases, you can literally find hundreds and hundreds of thousands of enzymes and predict what they might be doing. Okay, so this sets up the possibility of putting these together in non-natural combinations, which I want to talk about for the next few minutes. The other point is that very, very small changes in these structures can have very big impacts in activity. So you can completely change the enzymatic target by putting on a methyl group. You can completely change the profile of products that are made by a biosynthetic pathway by adding one new biosynthetic enzyme to it. Okay, so there's tremendous potential here. So the game-changing developments, really are that now there's hundreds of such enzymes. They don't exist as physical entities, but we can synthesize them. And the reason we can synthesize them is that the cost of DNA synthesis is now down to around 8 or 9 cents a base for, at least for academic labs. So it's feasible to think about making many of these and putting them together in non-natural combinations. And so that's what we're doing in budding yeast, essentially using the yeast cell as a microfactory to make new compounds. So this is our workflow. We synthesize the genes. We put them into combinatorially expanded YAC, so we can typically make YAC libraries of millions to even billions of clones if we wish, and we can screen those libraries in very high throughput, characterize the YACs that appear to be making bioactive products and purify those products and figure out what they are, and then move those hits into the drug discovery pipeline. So for the sake of time, I'm not going to go into the various assays that we're able to run with these compounds, but we've surveyed basically all of the major classes of natural products. We've made over 2,000 different genes across these different classes. We've developed essentially a custom YAC system. We use homologous recombination in yeast to zip these genes up into these libraries, so we can make many, many different libraries. We can show that the assembly fidelity is very high. It's over 90% using this system. And then what we do is use a trick of yeast genetics. We actually mate in our selectable reporters against any target of interest to the library, and we can simply plate. So we can get millions of clones on a plate, look for the ones that grow, and those guys are the candidate synthetic natural products that, that will be hitting the target that we've built into our assays. Okay, so I think I'm running out of time here, so I'm just going to very quickly flip through one example. This natural product is an alkaloid called violacin. It's sort of one of the standard products that's being made in yeast many times over. Here's our YAC, so the core violacin pathway, and then we loaded it up with all of these different diversifying enzymes. And the reason I chose this example is you can look this plate of the library, look at all these different colors. Okay, the violacin parent clone is a sort of dark black color, but then you see all these different colors. We screened those in very simple Halobased assays against a couple of different bacterial pathogens, including a model for TB, M. smegmatis. You can see this little halo here. And what we can show is that the clones from the library are making a whole slew of different products compared to the core pathway itself and compared to the natural producer. And we can essentially use metabolomic profiling to establish the relationships between all of these different peaks. And the bottom line is that these library clones are very different from the input clone, and they're different from each other. And here's just a clustergram to show that. Okay, so I'm over time already, and the idea is that we can make many different SynMP libraries. We can run them against many different targets and try and build this huge matrix of chemical genetic interactions. And we're starting to direct this process using this metabolomic profiling. So I think I'm out of time. I'm just going to skip to the end and just mention that we're also trying to now use a commensal yeast called Saccharomyces boulardii, which is a normal resident of the gut microbiome, to produce things like synthetic nanobodies and potentially even natural products, synthetic natural products, and other bioactive compounds as a way to try to begin to remediate the microbiome in various disease states. So we're trying to extend this idea of using yeast as basically a bioproduction system that we could actually move in situ into humans. And I think if I want to look 20 or 30 years out into sort of total insanity, that's where we're headed. Okay, so anyway, I wanted to end with a couple of key quotes here. I'm surprised this one didn't get put up yet from Yogi Berra. It's tough to make predictions, especially about the future. The future ain't what it used to be. So from the ridiculous to the sublime, from Lee Hood, don't underestimate the power of your vision to change the world. Whether that world is your office, your community, an industry, or a global movement, you need to have a core belief that what you contribute can fundamentally change the paradigm of thinking about problems. And I think that's a good place for me to end my talk on. And we're hoping that the kind of technologies we're trying to build will link into some of the efforts that Lee and Nathan and others are undertaking. So thanks very much to the funders and everyone who's contributed. And thank you.